Assalamualaikum and hello to the examiners. My name is Muhammad HILMI bin Muhammad Idris. My matrix number is 72420. The title of my project is The Growth Profile of Selected Commercial Baker's Yeast as cerevisiae in Ethanol Fermentation. My project is supervised by Dr. DAYANG SALWANI, BINTI AWANG ADENI. For the introduction, Saccharomyces cerevisiae is a type of yeast species widely used in baking, winemaking, and brewing since ancient times. Saccharomyces cerevisiae originated from the Latinized Greek words saccaro meaning sugar fungus, while cerevisiae means beer. Saccharomyces cerevisiae is believed in the 6th millennium BC to be used in winemaking, and it is one of the early processes. It is one of the early yeast used in winemaking. It is commonly used in biological studies as a eukaryotic model organism due to its properties, which makes it easy to be cultured. In baking, since ancient times, this yeast has a principal task of degrading simple sugars, inhibiting flour dough. This yeast also plays another role of forming beneficial secondary products of fermentation such as ethanol. The problem statement of this project is the rate of yeast fermentation and carbon dioxide formation has been demonstrated to be affected by glucose concentration. So a question has been triggered: will increasing glucose concentration affect the rate of yeast fermentation and carbon dioxide production? This question will be answered by this project. The objective for this project is to determine the growth performance of selected commercial baker's yeast using two different glucose concentration. And the second objective is to compare overall ethanol fermentability of selected commercial baker's yeast in two different glucose concentration. For the literature review, 2.1 Saccharomyces cerevisiae. Saccharomyces cerevisiae is a well-known yeast product which are used in baking and other industries. As stated in the introduction, Saccharomyces cerevisiae originated from the Latinized Greek words which means sugar fungus used in winemaking. Saccharomyces cerevisiae commonly utilized in biological studies as a eukaryotic model organism, according to Feldman in 2011. According to Feldman also, Saccharomyces cerevisiae under the microscope shaped like a spherically and also reproduce through budding mechanism. Saccharomyces cerevisiae has become necessary to human civilization due to its usage in beverage and food fermentation. The European yeast industry approximately produces around 1 million tons of Saccharomyces cerevisiae or baker's yeast annually, and 30% of them are shipped globally, according to Parapauli and his fellow researchers in 2020. 2.2 Baker's yeast production through fermentation process. Fat batch fermentation process was utilized in baker's yeast production according to Jelinas in 2014. Fat batch fermentation is a process where nutrient is gradually added into the fermentation media. For the process, firstly, the purest and strongest microbial starters must be seeded into fermentation tanks, and secondly, those microorganisms are kept under strict environments to maximize gassing power and also yeast biomass, according to Jelinas in 2012. 2.3 Growth rate of Saccharomyces cerevisiae. Measurement of Saccharomyces cerevisiae growth is usually done using three types of methods, according to Oliveres Marin and his fellow researchers in 2018. The methods are spot testing via serial dilutions, counting of colony forming unit CFU, and drawing growth curves. 2.4 Ethanol production. Ethanol is produced through the process of glycolysis. In glycolysis, glucose is reduced into two molecules of pyruvate and also produce two molecules of ATP and two molecules of NADH. pyruvate is then converted into ethanol, which are later further reduced into becoming ethanol. 2.5 Saccharomyces cerevisiae application in bread industry. The commonly used yeast in bread industry is Saccharomyces cerevisiae, also known as baker's yeast, which in the late 1800s are acquired from the remnants of beer manufacturing process. In bread making, the yeast cells absorb oxygen from the air trapped inside the dough, and under the anaerobic states, yeast cell development slows down and the fermentation reaction occurs and ethanol are produced. 2.6 Saccharomyces cerevisiae application in wine industry. In the 1860s, Louis Pasteur first discovered the unseen world of his activity during winemaking and fermentation process. Due to this discovery, Müller-Thurgau came up with the controlled wine fermentation process using starter cultures in 1890, which revolutionized wine business and became the cause of improvement in wine quality. For the methods and materials, first, preparation of yeast. 3 selected commercial baker's yeast will be purchased from the local market. The most commonly known commercial baker's yeast is the Mauripan. Yeast will then be cultured by inoculating 1 gram of yeast grains into a sterile flask with yeast malt broth YNB medium. The culture will then be streaked on a potato dextrose agar plates and incubated under 37 degrees Celsius for 24 hours. The single colony formed from the culture will then be stored in the refrigerator. Second, preparation of inoculum. 3 selected commercial baker's yeast will be cultured and transferred into inoculum media consisting of 5 gram per liter yeast extract and 20 gram per liter glucose. Inoculum is a place where the yeast cells will learn on how to adapt to the environment of fermentation media, which will be created in the next step. The inoculum needs to be incubated under the following parameters. pH needs to be 5.5, the temperature needs to be 30 degrees Celsius, and the times needs to be 9 to 10 hours. The 10 milliliter of the inoculum media will then be removed and centrifuged. Third, bakers use biomass production via batch fermentation process. The fermentation media will consist of the following items: 5 gram per liter yeast, 100 and 250 gram per liter glucose, 200 milliliter working volume or water volume. The media will be incubated under the following parameters: 37 degrees Celsius, for 48 hours, and under 150 RPM. 10 milliliter of the sample will be collected every 6 hours. Fourth, growth profile. Growth profile will be measured using dry cell weight DCW. Samples will be collected from the fermentation media and centrifuged at 10,000 RPM under 4 degrees Celsius. The supernatant obtained from the fermentation process are placed in dry falcon tubes for analysis. Fifth, glucose and ethanol analysis. Glucose and ethanol concentration will be determined using high-performance liquid chromatography or HPLC. Samples will be placed in tubes, centrifuged, and filtered using 0.2 micrometers Puradis filter membrane. The mobile phase used in the HPLC consists of 5 millimolar sulfuric acid with a flow rate set at 0.8 milliliter per minute. 6. And the last step, statistical analysis. All the data obtained will be calculated for mean and standard deviation using one-way ANOVA. Significant difference between treatments will be obtained if p-value is less than 0.05. For the expected outcome, first, the growth performance of selected commercial baker's yeast using 2 different glucose concentration can be determined. The selected commercial baker's yeast are expected to have 3 growth phases, which are lag phase, exponential growth phase, and stationary phase. Second, overall ethanol fermentability can also be compared between the selected commercial baker's yeast in 2 different glucose concentration. Here are the proposal schedule for this project. And here are the references used in this project. That's all from me. Thank you for listening.